Hi everyone, good evening. And today we're going to be talking about DNA, and we're specifically going to be talking about the structure and role of DNA of nucleic acids. Now, today's class obviously is going to be a little bit different because I'm hosting this on YouTube for you all. It's just because I was unable to have class today, right, due to some unforeseen circumstances. So I'm going to leave this work up for you, and I'll link a question sheet below, a Google form, that you can fill in all your questions, and I'll address them for you all by the next class. Okay. Of course, you can always feel free to stop, rewind, play over, and try your best to understand whatever is going on accordingly. If you still don't understand and you place it as a question, you know it's not going to be a problem. We'll go over it together later. Now, moving in to this topic, for those of you all who don't know, this is the start of Module 2, right? And we're going to be talking about DNA. Specifically, what we deal with in this chapter is DNA. Now, DNA stands for deoxyribonucleic, nucleic acid, right? Now, the deoxyribo part indicates that you're talking about a sugar. Specifically, you're talking about a deoxyribose sugar. Now, the word could even be further broken up to show that deoxy means that it's missing an oxygen. But let's look at the structure of it, and we'll see how that comes about, right? Now, DNA is actually comprised of 3 separate structures. Right? So you have, well, number 1, the first thing that we talked about is the deoxyribose sugar. Okay, and your deoxyribose sugar looks like this. So ribose, okay, looks like that. You have a carbon, H2OH, right? Okay, so this is your deoxyribose sugar. So if you weren't sure, it's actually missing the oxygen from this point here. This is why I didn't fill it in initially. What you'll notice at this point is that you have a hydrogen, just like everything else, you've got a hydrogen there, right? As compared to if it was normal ribose, you'd have had an OH or a hydroxyl group there. So for comparison, this is what ribose would look like. It's practically the same, except that down here, you'll have a OH instead of just the H. Okay, so this is an example of ribose. Now I'm giving you the structure of it anyway, even though it's not a component. Deoxyribose is the component that we need, right? So even though it's the deoxyribose that we need, I'm giving you the structure of ribose one time because later on we're going to be talking about RNA. And RNA is very similar to DNA except for a few key differences. One of them being that instead of using a deoxyribose sugar, it's using a ribose sugar. So instead of deoxyribose, ribose. So that's why I'm going to talk about RNA later on, and that's why I'm giving you the structure now. Okay. So if you didn't know and you couldn't guess it, if DNA stands for deoxyribonucleic acid, then RNA stands for ribonucleic acid, right? Now, if we were to look at it, right, just by the way, we can also consider the sizes, right? We can also consider the sizes, sorry, the numbers of the carbons. So this is carbon number 1 when looking at a ring structure. The first one away from the oxygen group. So this is carbon number 1, number 2, 3, 4, 5, right? So you can see that's a 5 carbon sugar as we expected, okay? All right, so this is the first component, it's the deoxyribose sugar. The next component is the phosphate group. So you need a phosphate group or a phosphate molecule. Okay, so this is your second component of DNA, the phosphate group. Now, the phosphate group is a very, very hydrophilic, right, ionized group that contains a phosphate central atom. So you have phosphate, oxygen, oxygen, believe it or not, oxygen, and I'm sure you could guess the last one, it's, yep, oxygen, right? So we have our phosphate molecule. And then the third thing that we need, it's nothing more remarkable than that. That's literally just the phosphate molecule. And then the last thing that we have is called an organic base. Right? So the last thing is an organic base. Now, your organic base, actually, you could have two types of organic bases, right? So your organic bases can be of two types. All right. So you have purines. Okay, purines, which are a double-ringed structure. They're known as a large organic base. And you have the other one, which is called pyrimidines. Right, a pyrimidine. This is a single ring structure. And therefore, by comparison, it's small. Okay, so you have your purines and you have your pyrimidines. Now, to look at the structures a little bit more closely, your purines, for example, they would look like this. So you have your purines, right? And they have this shape. Right? Like I was telling you, there are two types, right? So then the other one is the pyrimidines. And the pyrimidines are single ring structure. So it's only the first ring. And these are pyrimidines. Now, usually for ease, when we're drawing these diagrams, we tend to represent either one of them as just a rectangle, right? Either one of them is usually represented as just a rectangle. Okay. Now, on top of that, we have two of each type, two purines and two pyrimidines. Okay. There's a third pyrimidine, which we'll deal with when we come to RNA. Okay, but for now, the two that you need to know for purines, well, the two that they are, right? For the purines, we have adenine and we have guanine. And the two for the pyrimidines are the thymine and cytosine. Now, it's actually no coincidence that I wrote it in this particular way, because there are a couple of reasons why. So first off, trying to remember which ones are purines and pyrimidines can sometimes be confusing, okay? So I try to tell people little ways to try and remember it, because what happens is that they will bring questions for you, either in multiple choice or in paper 2. I mean, if it's a multiple choice question, obviously it's just 1 mark, right? But you don't want to be giving away these marks so easily. So they will bring something, for example, like a ring structure, and they will tell you, hey, well, is this a purine or is it a pyrimidine? So you'd have to look and see if it's double ringed or if it's single ringed. Then you'd know if it's a purine or pyrimidine. Then there are other questions that they'll ask you, well, is it adenine or guanine or thymine or cytosine? It only sounds tricky, but it really isn't. There are different ways that you can figure out which one is which. For example, by looking at the number of bonds that each one forms. Now, when we get to the when we get to the bonding, which is like literally the next section, I'll show you how you can count the bonds and figure out which one is which. But for now, this is the part that you need to know. What are the purines and what are the pyrimidines? So the purines are adenine and guanine. I like to remember it like saying, okay, you know what? AG, like Attorney General, it's a big position in the country, right? And well, there's no other way to remember this one, right? It's obviously the other pair, so they're small. Okay, so if you know that AG is big, then CT would definitely be the small ones. So you'd know if it's purines or pyrimidines. Okay, that's just a way I do to remember it. If it helps you, great. If it doesn't, and you have a different method, I'm all ears. I'm glad to hear it. Okay. So these are the three components together: the organic base, the phosphate group, and the deoxyribose sugar. Now, when you put all three of them together, you end up producing the nucleotide. Okay, so for the nucleotide, the deoxyribose, the deoxyribose, the deoxyribose sugar forms the basic unit to which everything attaches or bonds, right? The phosphate group attaches to carbon 5, right? And the organic base attaches to carbon 1. Okay, let me get the colors back. It was purple, blue, red. Okay, cool. So the deoxyribose sugar, right? The organic base and the phosphate group, right? And now you'll know which one each bonds to. So the simplistic diagram, right? The simple diagram of a nucleotide looks like this. Now the phosphates are usually represented in a circle. Sometimes you'll see the circle with the P, and sometimes you'll see it without the P. It all depends on the diagram that you're looking at and what the illustrator used, right? But it still represents the same thing. We can look back again at our carbon atoms. Number 1 would have been here, 2, 3, 4, and then carbon number 5 is out there, right? So you can see all of them. This, of course, here being your organic base, represented as that rectangle. And of course, the sugar in the middle is not just ribose, but it's your deoxyribose. Okay, and this is the structure of a nucleotide. Now, nucleotides are your building blocks, right? So we made our nucleotide using the deoxyribose, using the base, and using phosphate. The 3 of them together is what makes a nucleotide, and then the nucleotides form your DNA strands, right? Now, DNA has to be precise. When you think about the nature of DNA, it has to be precise, right? So if it's not precise, you end up with what we call mutations. And the mutations might be bad, right? It could be good too. Mutations can sometimes be good, but mutations are often bad also. Okay, more often than not, mutations are actually bad. Most mutations tend to be detrimental to life, and it will phase out because the organism won't survive or won't even be able to survive long enough to pass it on. But we get to that in a future chapter, right? So the point is that we want to ensure that DNA is stable, and we want to ensure that it's accurate. So when it replicates, when it does whatever it has to do, it retains the same information. All the time, you want to make sure we retain the same information, right? So with your deoxyribose, all right? We're going to look first at complementary base pairing and see how that influences DNA. And we're going to learn about the DNA double helix as well also. All right, so we're going to talk about complementary base pairing. Okay. All right, so first up, the same thing I was telling you is that DNA has to have or has to be a very stable structure so as to preserve the information it carries. Changes in DNA mean changes in proteins right? So the changes in DNA could be disadvantageous to them, okay? Now, in order to do this, right, DNA actually has to exist as a double strand, right? It actually has 2 strands of when you look at DNA, right? So DNA will exist as a double strand antiparallel helix. Okay, exists a double-stranded antiparallel helix. Okay, so all right. So what do those words mean and stuff, right? So we have to still talk about the complementary base pairing itself, right? But you know, we have a couple other things to talk about with it. Okay, now first up, before we go and start tackling all of this, let's start talking about the double-stranded aspect of it, right? And then we'll talk about the antiparallel aspect of it after. Now, for the double-stranded, right? So DNA, so the DNA double helix, okay? So first up, to form a single strand of DNA, right? To form a single strand of DNA, each nucleotide needs to bond to each other. Right? So we need to get each nucleotide to bond to each other. So this structure, this nucleotide, needs to bond to other nucleotides to be able to form the single strand. Okay? So where does this bond occur, right? Now, the bond occurs right? The bond occurs when the phosphates attached to the carbon 5 of one nucleotide right, of one nucleotide undergoes a condensation reaction with the hydroxyl, which is, if you could remember, as the OH group, right, with OH group of a carbon 3 on another molecule, well, another nucleotide. So off the bat, you could tell that the two carbons involved are carbon 5 and carbon 3, right? Okay. The name of the bond, right, the resulting bond anyway, is a phosphodiester bond. Okay, it's called a phosphodiester bond. And it looks like this. So you have your nucleotide. Just going to put a tilde there, right? And from here, you'd have had your OH group sticking out, right? That's because that's your carbon number 3, carbon number 1, 2, 3. Just 4 and 5 is going to put it in there anyway, right? My OCD would kill me if I didn't. And let's look at the one below, which I guess I could draw it with a different color so you could see one from the next, right? You have, let's see if I can place this correctly. Oh. It's terrible. Yeah, that's gonna work. That's gonna work. I mean, it's a YouTube video, but you all already know that I couldn't draw from the beginning, right? Right. So you have from your carbon 5, you have your phosphate head. Sorry. Right. O negative. And let me just I might have to just move this down a little bit because I put it too close. Right, good. And then you have OH here as well. All right, this is your phosphate. This is the unionized form. I know I didn't put an OH in the first one when I made it for you all, but this is the unionized part on top. Now, the bond forms here. Oh, sorry, let me label it off. Carbon number 1, 2, 3, 4, and carbon number 5. So the bond that's going to happen now is going to be between this OH group here and this OH group below. Shouldn't use that color. Just use a green or something. Right, so the bond is going to form between here. All right. We're going to get water exiting, H2O, right? Which is water. Condensation reaction after all, right? And you end up forming this. Right? So this would be your phosphodiester bond. Since I wrote it in orange, let me stick to the orange. Right, so that would be a phosphodiester bond. And of course, if it's DNA and it's multi-stranded and it's quite a lot of these nucleotides all joining to each other, right, then phosphodiester bonds are going to keep forming from one to the next. Okay. All right. We can draw this in a more simplified version because, when you're doing this in exams and stuff and they want a more generalized version, you can do a simplified version. All right. So the simplified drawing would look something more like this. Right, and so on. Okay, that's what it looked like. Of course, you could put your P inside here for phosphate, but like I was telling you, you don't necessarily have to, right? Another thing to note when you have this multiple times over and over and over is that, the phosphate and the deoxyribose. Phosphate and deoxyribose, when they get bonded together, by the phosphodiester bonds, is collectively called a sugar phosphate backbone. Sugar phosphate backbone. Okay, just so when you see the lingo, you'll know what we're talking about, right? It's called a sugar phosphate backbone. So this is called the sugar phosphate backbone. All right. Okay. So with that in mind, let's talk about that antiparallel nature, right, as well. All right. And then we'll go back to talking about the actual complementary base pairing, right? Now, the antiparallel nature simply means that that they run in opposite directions from each other. Okay, that's all. That's pretty much all that it's really implying. Right? Okay. So the antiparallel nature, if you're writing and at the same time as me and now you're thinking it's looking bad, don't worry about it. You can just write this below because it's not really much anyway. Okay. All right. So the antiparallel nature, right? If you're wondering why I'm not extending and writing further out, it's because when I make the PDF for you all later on, if I draw too widely out, it squishes all the work together. So that's why I'm trying not to go too far to the right. Right? The antiparallel nature of DNA simply means that each strand runs in the opposite direction from the other. Right? Each strand runs in the opposite direction from each other. Okay. Okay. Right? So the antiparallel nature. So if you were to draw one strand like this, and I'm gonna have to see if I hate to draw this part of it because it's very confusing for me. But okay. So if you draw it like this, and for the sake of it, I'll draw a third one, because why make my life easy? Gosh, those lines are so crooked. It literally looks the same. Why does it look the same? All right. Anyway, whatever. Right. So yeah, you've got your lines, and they're gonna end here. Right. Of course. Right, for both of them. Then if you look at it, remember, I'm numbering these again because it's important in a little while. We have number 1, 2, 3, 4, and 5. Take a look here, number 1, 2, 3, 4, and 5. Those are our carbon numbers. Just labeling the top one and the bottom one because the one in the middle isn't really relevant to what I need to talk about later, but you can see that the numbering for that one in the middle is going to be the same anyway. All right, so let me draw it in the opposite direction now to show you what it looks like. Okay, so this is where it gets confusing a bit for me to draw it because I can't really draw very well upside down. Yeah, you see, getting confused already. Okay, good. And this one would be sticking out here. Phosphate. Usually we draw the phosphates a bit larger than the oxygen because it's obviously going to be larger than the oxygen. The phosphate group contains, you know, 3 or 4, well, 4 oxygens attached to it. So obviously it's going to be bigger than one single oxygen. That's why we could tell that this is an O and that one is a circle. Okay. Okay then. Right. Then we go to the next one. Okay. Probably a little bit too close or something. Okay, cool. Look at that. I'm really good at drawing, clearly. I cannot roll now. Okay, yeah. Oof. Spoke too soon. Spoke too soon. Okay. All right, that's the oxygen. Great. And then we have another one. Right, it's coming out here. And we'd have the, right, good. Okay, so if we were to label these oxygens, these carbons now, it would be carbon number 1, 2, 3, 4, 5, right? And the same thing would hold here, number 1, 2, 3, 4, 5, right? Nothing has changed about the labeling, despite me rotating it. The only thing is that I'm writing the numbers for us to look at it, and I'm not writing it upside down, okay? Like the rest of it. So this is what it means by talking about the anti-parallel nature, right, of the strands. They run in opposite directions from each other. Now, they're actually held together, right, by hydrogen bonds that occur here in between. I used green for the, right, okay, that's fine. So let me see, change to purple. Purple could work, all right? We have hydrogen bonds that occur here. Okay. All right, is the same thing that happen on this one. All right, so you have hydrogen bonds that can occur. All right, the hydrogen bonds hold them together. Okay, so hydrogen bonds occur between the organic bases holding each antiparallel strand to each other. Right, holding each antiparallel strand to each other. Okay. All right. Now, this is where the complementary base pairing thing comes into play, right? the complementary base pairing. Now, I think I used orange at the beginning, right? The number of hydrogen bonds that can form between the base pairs is specific, right? Okay. The number of hydrogen bonds that can form between the base pairs is specific. Adenine and thymine can both form 2 hydrogen bonds, right? So adenine and thymine could both form 2 hydrogen bonds, okay? And guanine and cytosine can both form 3 hydrogen bonds, right? They can both form 3 hydrogen bonds, okay? So if you notice something, right? These are both purines. Well, these are both pyrimidines. Right? Those are both purines and those are both pyrimidines. It means, right, that bonding between base pairs always occurs between one purine and one pyrimidine. Right? Between one purine and one pyrimidine. Okay. Further to this, if the bonds are to be stable, or if the bonding is to be stable, then adenine must bond to thymine. And guanine must bond to cytosine. Right? So the reason for that is because if adenine and thymine bond to each other, since they both only require 2 bonds, right? Since they both only require 2 bonds, then it means that they would both be satisfied, right? They would both be stable. And then if guanine and cytosine bond, they both require 3 hydrogen bonds, so they'll both be satisfied and then they'll both be stable. Okay? Makes sense, right? Okay, so I don't know why I was waiting for an answer there. I forgot that is a recording, clearly, right? So this is how I remember it. Okay, like I told you all before, I like to try to give you all ways to remember it. I remember it like this: adenine and thymine could bond together. In terms of the letters are both straight edged, and guanine will bond to cytosine. Draw that a little bit better. Guanine will bond to cytosine. And this one is because both the letters are curved. Okay, so that's just the way I remember it, and I draw it with the bonds so I know how much each bond is: 2 for AT and 3 for GC. right? Just a little by the way scenario. Okay. Okay. So this is how we get to that whole concept of complementary base pairing, and how we get E is always bonded to T's, sorry, and G is always bonded to C's. Okay. All right. So now I have a couple more things to write about this, which I will be doing shortly. In any case. Okay. All right. So I just wanted to make sure you're okay with that so far, and let's move forward, right? Now, the next thing is about this DNA double strand, the antiparallel nature again, right? We're going to be talking about that one more time. Okay. All right. So we have double helix and right, good. Okay, cool. Now, let me see if I can copy paste this whole thing. All right, cool. Okay, good. Naming of the strands. Okay. So each antiparallel strand is labeled based on, right? So each antiparallel strand is labeled based on the carbon atom closest to its end. All right. Ends, sorry, ends. Okay. Right. It is also shown in terms of its direction. Right. So it's also shown in terms of its direction. Right. Using an arrow. To show the carbon furthest. Now this, it just looks more complicated than it is, right? It really isn't that complicated. So this is what it looks like, right? If you look at this strand here, the black strand, if we're looking at the black strand, at the top, right? The closest carbon to the top is a 5 carbon, a carbon number 5. So we call this end the 5' end of this chain, the black chain, right? So by comparison, the lowest carbon, the one that's closest to the end, like the absolute end of the other chain, is a carbon number 3. So we'll call this the 3' end. All right. And considering that we're looking from top to bottom, it looks like it's 5' to 3'. So we would label off this black strand, right? So for example, the black strand of DNA has a carbon 5 at the top and a carbon 3 at the bottom. Okay. So this strand will be called the 5' to 3' strand. Okay, simple as that. Let's do the same thing now for the blue strand. So for the blue strand, we can see that at the top, the closest one is the 3', right? Because that's a carbon 3. And the one closest to the bottom is a 5'. It's anti-parallel after all. So you would expect that the numbers would have been inversed because the strands are actually inverted to each other, right? So it's going from a 3' to a 5' strand from when you go top to bottom, right? So then that would mean that for the blue strand, so the blue strand of DNA has a carbon 3 at the top and a carbon 5, and a carbon 5 at the bottom. So this strand will be called the 3' to 5' strand. Okay, so they're just named based on the relative positions of their carbon 5s and carbon 3s. Which one is at the top, which one is at the bottom. And that's it. That's pretty much it. Okay. Okay. Right. So these are the two strands are named, right? So they go opposite each other and therefore their carbons are opposite each other. Now, some of the questions I might have is like, well, how do you know, should I start from the top to the bottom or from the bottom to the top? Because if I went from the bottom to the top, then this blue strand would have been, if I went from the bottom to the top, this would have been the 5 to 3 and this would have been the 3 to 5, right? Yeah, 100% agree with you. It can be confusing. As a matter of fact, what if they rotated it to 90 degrees and now it was, running in this direction? Okay, so my advice to use this, because of the nature of it, like it could be, up to down or left to right or right to left or down to up. Because of the nature of it, usually take it as if you are reading it on a book, right? We tend to read from left to right and from top to bottom. That's normally how we do it. So if they give you the strand in a vertical form, read it from top to bottom. If they give you the strand in a horizontal form, right, read it from left to right. That's how I usually tend to look at it. Now, once again, right, I know that that is relatively speaking subjective. So as a matter of fact, that's completely subjective. And it's true, it is. So here's the thing about it, why it would work out in an exam. Now, in an exam, if they bring the strand for you like this, they won't ask you a question like to label it unless they make it clear that one strand is something and one strand is the other. Okay. Otherwise, they'll have to accept both answers depending on if they tell you, just label it, or if they tell you label it from top to bottom. So they'll give you a clue as to what it is you should do. The only other way that I've seen this come, and they don't give you any information, like actually don't tell you what to do, is in a multiple choice. Now, usually what they do in a multiple choice is that they will say, okay, this is a strand of DNA, figure out which strand is the RNA equivalent of it. Now, when we go to RNA and stuff, you'll see how we get this. But basically, you just have to look at the code and see which one lines up in the correct way. Okay, does the code that they give you line up from top to bottom, or does it line up from bottom to top? And then you'll know which one it is. Once again, relatively subjective, but I have not seen questions like that from them in years, to be honest, without them being a little bit more explicit as to, okay, you know what, this is what we require of you. So I haven't seen questions that would worry you in terms of trying to figure out what direction these strands should be. Okay, I haven't seen anything like that, so I wouldn't worry about it too much if I were you. Just once you know which one is 5' to 3' and 3' to 5'. Either they tell you, or if they don't tell you, you could use the context clues of the question to figure it out. Okay, but we will be practicing some questions later on with it anyway. Okay, so nothing too much to worry about there. Okay, so with that in mind, right, and understanding the naming, then it's going to take us now to DNA replication. Okay, fun stuff, DNA replication. Okay, all right. Okay, now DNA replication is actually pretty simple. You just need to understand a few simple concepts, right? And it'll be easy to figure out all of these things. Now, the first one, right, is that to understand what we talked about before about DNA replication being successful, DNA needs to be accurate and not change. We talked about that in the beginning when we said that DNA has to be very stable and not incur changes. Changes would be mutations, right? So first off, before we start talking about about DNA replication, we need to start understanding that for successful DNA replication, right? For successful DNA replication, the code, right? The code on the DNA right, needs to be accurately replicated from the old strands to the new strands. Right? So the old strands are like your parent strands and the new strands are like the daughter strands, right? That's effectively what they're saying. Now, because of complementary base pairing, right? And we called it orange because of complementary base pairing. Right? Bases can only pair with their, well, you know, it's hard to use the same word, but with their specific complementary opposite base, right? I.e., well, I mean, this is sort of repetitive, but A bonds to T, and C, well, G, right, bonds to C. it doesn't matter if I write C bonds to G or G bonds to C, but I'm just trying to keep it in a way to show you all the purines first and the pyrimidines second. So I just didn't want to break the continuity, right? That's all. It doesn't matter if you write A bonds to T or T bonds to A, it's the same thing. All right. So i.e. A bonds to T and G bonds to C. So how does that show, like how does that enable complementary base pairing, right? How does this enable accurate DNA replication? Okay. Because DNA replicates by semi-conservative replication. Okay, semi-conservative replication. So if you're wondering what semi-conservative replication is, no problem, right? Semi-conservative replication basically means that your DNA helix is going to split and each strand becomes a template for a new strand. That's what it means, right? Each strand becomes a template for a new strand. Okay, so semi-conservative replication. This means that the DNA, right? This means that the DNA double helix will unwind split, and each new strand, sorry, and each old strand will act as a template for the formation of a new strand. Okay. So one more time, this means, or should I say therefore, when DNA replicates, the replicated how should I write this? When DNA replicates, the new double helices are comprised of one old strand as well as one new strand. Okay, as with everything, diagrams tend to make it easier. So if we were to look at this, for example, right, let's say that we have our strand and it looks like this, right? So we're going to make our double helix, right? That's one strand. Let's call this our 5' to 3' strand. All right, and we can use a color that would stand out next time. How about the green? Right? And the next strand would be the antiparallel strand that also wraps it around the same corners, but therefore it's antiparallel. This would be a 3' to 5' strand. Okay, so this is your original DNA double helix, right? That's the original DNA double helix. Okay. So this would be like before replication. Probably write that on this side actually. Right. So this is before replication. All right. And then it's going to unravel like that. All right. So this would be like during replication, right? It will unravel. And after replication, you'll end up with right. So you would end up with the orange on this side. Which is 3 prime. I'll write in that after. And the green one on this side. All right. And our 2 new strands are going to form against them. So you have 1, 2. Right. So if your original green strand, right, was the 3 prime to 5 prime, then your blue strand is going to be a new 5 prime to 3 prime. Okay. And that means that with respect to the orange side, right, if this is your 5 prime to 3 prime, then the blue is going to be your new 3 prime to 5 prime. Okay, so each one consists of a new strand as well as an old strand. Okay, so by the end, right, by the end of replication, right, each old strand right, pairs with a complementary new strand. Okay, so this means that the old, sorry, let me write that a little bit differently, save myself some time. This means that the old 5 prime, the 3 prime strand has a new 3 prime to 5 prime strand. And vice versa. Right? So for the green one, the 3 prime to 5 prime strand has a new 5 prime to 3 prime strand. Okay? Right? So they get new strands. Now, here's the thing about it, right? Remember, the DNA is replicated accurately. So in terms of the messages, like the codes that they have, the old 5 prime to 3 prime strand, the old one, is like the worst thing to highlight it by. The old one is the exact same code as the new one. And similarly, the old 3 prime to 5 prime strand is code for code the exact same thing as the new 3 prime to 5 prime strand. Okay, there should be, if DNA replication was accurate and there were no errors, there should be exactly the same thing. Okay, so if we were to like pretend and make up a little code for it, right? Let's just say that our original strand, we just took a little section of it, right? This is our 5' to 3', right? And the green, 3' to 5', right? Let's say we just made up a little code for it, 2, 3, 4, 5, right? Let's say A, C, T, C, G, all right? Then on the opposite side, you'd have to have pairs that line up with this. They'd have to be the equivalent. A's always bond to T's, C's always bond to G's. C, G, G, C, right? If this is the code that we have, then when we split it up, right, and they break apart, you'll end up with this. Right, so you end up with that, and the new strands are going to form against them. So a new strand forms here. This is going to be a new 3' to 5' strand. What is going to pair? So if we look at what pairs, you'd see T has the bond to A, G has the bond to C, A has the bond to T, G to C, and then C to G. Complementary base pairing, right? Look at the code of the blue strand. The new strand is a 3' to 5', and the code is ACTCG. What was the code of the original green strand? ACTCG. It's literally the same code. So what do you expect is going to happen on this side now when the new 5' to 3' strand forms? Right? You're going to get the A's bonding to T's, C's bonding to G's, T bonding to A, C bonding to G, G bonding to C. Look at the strand 5' to 3', new blue. TGA, GC. Look at the original orange. TGA, GC. So it's the same code that's being written over and over. So this is how come the information is conserved, because you're literally carrying back the same information. Okay. Right. So this is how DNA replication occurs. Now, we have some more to talk about with DNA replication, like, for example, the enzymes that are involved. Okay. This is a bit of a longer note because you need to know what each enzyme does. Okay. And how it accomplishes such things, right? Such wonderful things. So, right, I will, let's see, should I do this today? Right. Okay. So the 3 enzymes, right? So we have 3 enzymes in particular that we need to talk about when we talk about DNA replication, okay? So DNA replication continued. Okay. So DNA replication primarily uses 3 different enzymes. Okay, so the first one is called DNA helicase. The second one is known as DNA polymerase. And the third one is known as DNA ligase. Now, before you go off and run around on the internet and look for all these different processes, right? They are correct. These are the 3 enzymes. but these are generalized categories. So sometimes, depending on what video you're looking at, you'll hear them talking about DNA polymerase 1 and DNA polymerase 3, right? You'll hear them talking about different types of ligases and other enzymes, including one called a primase, which I'll talk to you all about just now also, right? So there are more specific names for these enzymes, but these are generally speaking the ones that we'll be referring to. So like in your syllabus, for example, the use of polymerase 1 and 3 is clumped together as just DNA polymerase. Okay, so it's like that. All right, so let me just mention it one time, right? Also, another enzyme involved is primase. The enzyme, right? The enzyme is responsible for adding a short sequence of RNA, and this is very important. Careful whenever you're writing this up. It's RNA, right? Adds a short sequence of RNA to the exposed unzipped DNA. All right. The segment of RNA that is added is known as a primer. Okay, so the point of the primer, by the way, is two things really. The first thing is that you see unzipping DNA, like how we've been looking at it so far. Like if the whole thing gets unzipped, that's a little bit inaccurate. But it's simple representation, and this is a drawing and not a graphic, right? Like not a moving graphic or video. So I had to draw it. Now, if you try to unzip the entire DNA all at once, then unfortunately that would mean that the DNA would become unstable and it could potentially react to things on the outside. We don't want that. So the primer, right? What the primer does is that the primer will attach as soon as the DNA becomes unraveled, as it becomes unzipped, and it stabilizes the DNA, right, in that region, and it provides a place for attachment of the DNA polymerase. You remember these things are enzymes, and because they're enzymes, it means that you have to you have to have a specific shape, right? Like they are enzymes after all, right? You have to have a active site and a substrate that matches. Well, DNA polymerase, in order for it to attach to the DNA, needs that primer there. Otherwise, it won't attach. So the primer is necessary mainly for two things, right? One is that it stabilizes the DNA, and the second thing is that it's a site for attachment of the DNA polymerase. Okay? The two functions of the primer are, so number one, stabilizes the DNA when it is unzipped, right? And two, right, provides a point for attachment of DNA polymerase. Okay, now the thing about it, right, with this DNA polymerase, it sounds like if the primer has to be added and then the polymerase has to be added, right? They kind of happen all at the same time. Okay, so it's not like one has to be there and then the other one has to seek it out. It sort of is like that, but at the same time it happens so quickly. It's kind of like if they all happen all at the same time. Okay, so the primers go and attach at the same time as the polymerase. Okay, that's effectively how it goes. All right. Now we had to talk about the actual process, right? This is where it can be a little bit tricky. Okay. Um, I'm wondering, I think we should probably stop here and I'll go through that process with Jill because I know you all will have questions about it when we meet up. So I'm thinking that it might be best if we stop here and we'll go through the actual process when I see you all next time. Okay. No, it's not a hard process, but I feel like it can get lost in translation if I'm not there to take your questions at the same time. But what I'll do is I will link this whiteboard one time. I'll link it immediately for you to follow. And I will link another whiteboard, or at least I'll add the image to this one, if anything, that includes the process so that you can read it out, look at it, and you can take that in consideration for our next class as well. Okay, so this is not necessarily going to be a normal thing where I'll teach classes to you all like this. I hope that you understand, and please don't forget that in the link below, in the description below, I'll leave a link for you all to fill out the Google form if you all have any questions about anything in particular. Actually, also, I'm hoping that you all could put in some comments, like, if you thought it was good or bad, was this helpful or not? I really would like to know what your feedback is on something like this too. Should I create more content like this for my YouTube page as well too? I'm just curious to know your feedback as well too. So I'm going to leave the link in the description below. Please feel free, please fill it out, right? And kind of just, even if you don't have any questions, just fill it out and let me know that, hey, I finished the whole video, it was good or bad, or I understood everything. So I just kind of know where you all are at for next week's class. If you all want to share this video with other people, if you think that's good enough and you want them to watch it too, your friends in school struggling, I think that's fine, whatever, that's no problem with me. It is on YouTube after all, and it's probably going to stay there. Okay, so I hope this was good. I will see you all next week. And like I said, please don't forget to fill out this stuff below. Okay, take care, everyone. Bye.